video, we are going to talk about the significance of the Avery MacLeod and McCarty experiment. In order to do so, we must quickly look back at the experiment completed in 1928 by Fredrich Griffith. Griffith, using mice, demonstrated that cells can transform. However, the mechanism by which this process occurred was not identified. He showed that rough cells can transform into smooth cells, but what molecule is being transferred to cells enabling them to be transformed? That was the major question posed by Avery MacLeod and McCarty in 1944. Experiments by these three built off of Griffith's experiments, providing evidence that DNA was most likely the molecule being transferred during transformation, enabling cells to transform. In the Avery MacLeod and McCarty experiment, they utilized heat-killed smooth virulent Streptococcus pneumoniae to determine which component of those cells was the transforming molecule. To determine what exactly the transforming substance or molecule was, they performed a series of treatments to try and destroy or inactivate the transforming molecules within their killed smooth bacterial sample. If they were able to destroy or inactivate this transforming molecule, it would inhibit its ability to be transferred to rough cells, and thus transforming them into smooth cells. Remember, the starting material of these experiments were killed smooth bacterial components. In the first experiment, they treated the sample with ribonucleases, which degrades RNA. Thus, in this sample, all the RNA is gone or inactivated. They then incubated their sample with a live rough bacteria and assessed for transformation by injecting it back into the mouse. And what they saw was that these live rough bacteria did indeed transform into smooth virulent bacteria, suggesting that RNA is not the transforming molecule. In the second experiment, they treated the sample with proteases, which degrades proteins. Thus, in this sample, no proteins remain. They then incubated the sample with live rough bacteria and showed that the cells were able to transform into smooth virulent cells, showing that proteins are not transforming molecules. They then repeated the same experiment with lipases, which degrade lipids, and again showed that lipids are not the transforming molecule, as the live rough bacteria were able to be transformed. The final experiment treated the sample with DNA-specific nucleases, or DNAase enzymes. These enzymes degrade DNA, thus these samples do not contain any DNA. After incubating the live rough Streptococcus pneumoniae with this DNAase treated sample, the cells did not transform, and this suggested that DNA was a transforming molecule. The Avery MacLeod and McCarty experiments demonstrated that DNA is most likely a transforming molecule. However, this experiment did not directly show that DNA was the molecule that was transformed into rough cells and then transforming them into smooth cells, or nonvirulent to virulent. Even though they did not show direct transfer of DNA into bacterial cells causing transformation, that would later be shown by Hershey and Chase and in their experiments in 1952.